questions, but I'll start us off with a bit of background. I've asked Lori to give us a flavor for what are biomarkers, what do they measure, and what's different between a biomarker and a companion diagnostic.
Okay, great. It's a great question, and anyone on this panel could talk about this for hours on end. I'm going to try and do this in just a few sentences. So in the world of everything biomarker, although that's a global term, what we're going to be talking about specifically here today is a really defined subset of types of biomarkers. If you look at biomarkers in general, there's about three primary categories that people talk about. The largest of those are what are called mechanistic or pharmacological biomarkers. They are things that actually allow you to understand the engagement of your compound with your target. And they're used in early development in industry quite frequently. In fact, some companies require that every program have some kind of pharmacological biomarker available before they progress that project into full development. And what's important to know about that is the endpoint that you're looking at as a biomarker may or may not be directly relevant to the disease in question. It is simply an indication that there is actual engagement at the target in terms of inducing some kind of pharmacological action. You then move to the next step, which are outcome biomarkers. Now, these are much harder to get. These are things that are surrogate endpoints that give you an earlier read in a clinical population that allows you to predict whether or not that drug is actually going to be effective in the disease in question. They're much more rare. They're harder to come by. They're easier in some particular types of indication like oncology. In neuroscience, they're very hard to get, and everybody would love to have them, but there just aren't that many out there that are validated. From there, you move to the topic of interest here, which are more your patient selection markers. These are things that allow you to do a number of things in clinical trials, either enrich your patient sample by choosing patients that have a particular characteristic, stratifying your patient population, allowing you to power up your studies with the most relevant patients possible. Now, a subset of those types of markers, which allow you to choose those patients, is what then can be chosen and put forth as a companion diagnostic, because what they're actually going to do is be co-developed with your therapeutic and allow you to administer that drug to the patients who are likely to respond to it. And that's a much smaller, more select group. It's also much more complicated to develop those because they go through the very same processes or very parallel processes for development with government agencies. They're actually being co-developed with the therapeutic. So it's a much more complicated thing because they have commercial value as opposed to the other endpoints that I talked about that are usually used for internal decision making and not for marketing the drug.
Well, that was great. I think that's very helpful. And so, Cecilia and Lori, representing big companies, how many of your internal programs have biomarkers? And is it true for all therapeutic areas, CNS included? How about?
I was going to ask if you can want to pass the microphone so we don't drop any of the bottles, but. So we just announced that over 75% of AstraZeneca's internal programs have what we called adopted personalized healthcare strategy. Now, I need to define that very clearly. By that, what we mean is we're looking to the early biomarkers, as Laura just explained, all the way to the clinical practice. So we, in the last 3 years, went from 10 to 15% to pretty much where we're getting to 80% of the programs incorporating personalized healthcare strategy. What we also identify, those programs that have a personalized healthcare strategy will increase the success into moving through the different stages of development. I'm not going to say shorten the stage of development or making it less expensive. We can talk about that because I think it's a really good topic. There's a little bit of a myth behind it, but definitely it's increased the success rate in terms of the scientific basis for the drug development program.
The thing that I'll add to that, since you're talking about success, is there are a couple of issues when you're a drug development company you need to pay attention to. One is being able to successfully launch your product, which means you have to be able to show efficacy in the disease. The other is the commercial aspect, because of course drug development companies are businesses, so they're looking to actually be able to bring in revenue from their product. One thing that comes up often that makes companies a little concerned about getting into using diagnostics, companion diagnostics, is it can limit or appear to limit commercially the value of your project. because you're now talking about only being able to use that product in a very select, smaller group of patients. One thing that you need to think about commercially, though, is whereas you may not be giving that product to as many patients, the likelihood of it working in the population where you're applying it is virtually 100%. So it's a trade-off. Those folks who will get it will respond 100% of the time, as opposed to a more homogeneous kind of toss it out to all patients approach.